This is going to be an introductory PowerPoint presentation, and the full sessions will be on your e-learning, the original lectures, and it is important that you go and study these lectures. The theme of this PowerPoint presentation is critical thinking. What is critical thinking? Critical thinking is that when I read a paragraph, I read a sentence, I stop and think and ask myself, is this usable? And then ask more questions about it. So it is a matter of questions and answers. Critical thinking is not accepting anything and memorizing it. It is about asking questions about any piece of information. We are about to start studying the very beginning of the human life. This is going to involve a story that has a beginning and then events well timed and well understood, and event after event after event till the end of the story. As we start talking about the story of the development of the human body, and that is science of embryology, we need to have a bird's eye view. You know what is a bird's eye view about certain facts that we are going to face in the next slides of this presentation. First of all, the human body is complex. It's very complex. It involves billions of cells organized in a perfect way to function very, very efficiently. All these things, they come out from a single cell. The human body takes time to build, and everybody knows that it takes around 9 months plus a few days, plus or minus. And the human body is the outcome of very delicate, very precise changes happening in the body. These little processes that will take place, they are in sequence, very prompt sequence. Everything finishes and then the next process begins. Sequence is very important, and the timing is also very important. If any process is exposed to delays, the development will either be defective or it stops. And this sequence of events is going to be discussed, and it is very important that we should pay attention to it. The study of development of the human body or any organism is called embryology, and it is part of science of medicine, and we are going to see few examples of that. Why should I study embryology? If you look at this image, you can see that around the mouth, especially the upper lip, the lower lip has developed well. Now, in the upper lip, you can see that the left nasal opening is not there. It has not developed. It started, but not complete. And you can see that the left half of the lip, upper lip, should come and meet in the middle, as well as the right lip. Now, the process of formation of normal, nice upper lip did not end well, and it has stopped at one stage or another, and the baby is born with something wrong went in his development. Here is another example. What is this situation? This is a widely opened mouth, and you can see that this is the tongue, these are the teeth, this is the hard palate, the bony part of the roof of the mouth, and within this rectangle you can see that what is supposed to happen is that these two halves, they come and meet in the midline. Well, they are formed and they went along the process and stopped. So there is a gap between the mouth and the nose. This is called cleft palate. What is this image? This image is a diagram of the heart, and if you look at this rectangle, you can see that there is a gap between the left atrium and the right atrium. This gap should not be there. What separates the two chambers should have completed and result in total separation. This process of total separation did not happen. It is stopped at certain stage, and the person is born with a congenital heart defect. That is why we need to understand embryology so that we can treat people. What is this? This is a kind of X-ray. It is called CT scan of the human body in what we call coronal section. Probably you know by now what is coronal section, and the area in the middle is the abdomen. And here you can see that this person has a left kidney. This is the left kidney, and normal people should have two kidneys. But if you look on the right, there is no kidney. If there is, there should be the same as the one on the left. So this person was born developing a kidney. The other one did not start. That is one of the congenital problems of forming the kidney. And there are so many examples. What is the starting point of formation of the human body? Here is a view of nearly the starting point. We have two gametes. One is a female gamete and the other is a male gamete. They should come together, and once the nuclei of these two structures are met and fused, then we will start having the starting point of creating a new life. Now, this large cell is the ovum. It is the female gamete. Sometimes gametes are called sex cells, and it is a single one. And these sperms are trying to fertilize or to join the nucleus of the ovum, but only one is going to succeed. Now, the ovum and the sperms, they need suitable environment to survive in. Before these gametes develop, we should prepare the place where these gametes are going to live. Now, what is this place or this environment? It is what is called the genital ridge. Genital means reproductive system, and the ridge means an elevation. And this is where the story begins. Let us see what this image is telling us. We ask a few questions. What is this structure? This is the future human being. It is the future embryo. Now, this is called the embryonic disc. The human body develops at its first stage as a disc, a flat, oval-shaped disc. Above the embryonic disc, we have a cavity called amniotic cavity. Don't worry about the word amniotic, because you are going to hear about it later on as well. This cavity is filled with fluid. Why with fluid? For the sake of nourishment of the embryonic disc. Now, under the embryonic disc, we have another sac, the yellow colored sac, and this is called the yolk sac. What is it filled with? It's a space filled with fluid, nutrient fluid for the embryonic disc. If we follow this arrow, we find that this yolk sac has an extension into the umbilical cord. This is called allantois. We will know shortly why we should know these things. And at the tail end, with the allantois projecting into this structure, at the tail end, this is going to form the umbilical cord, going from the umbilicus of the fetus later on to the placenta in attachment to the mother's uterus, so that it gets nourishment later on. This diagram is going to tell us a lot. So let's ask a few questions and try to explain. What is this red line? It is the midline between right and left halves of the embryonic disc. Remember, at early stages, the human body is a disc, an oval disc, just like a piece of paper. Now, at later stage, few days later, the one paper disc-like structures is going to be 3 papers, one on top of each other. The top one, the blue one here, is the ectoderm, the outer layer. Your skin is from ectoderm. And then we have the green colored structure, which is the middle paper of the 3, is called mesoderm. Your muscles and other structures under the skin, they develop from this middle layer, the mesoderm. Then we have the lower paper of the 3, the lower layer of the embryo is called an endoderm. For example, the lining of your stomach and intestine is developed from this endoderm. Now, this mesoderm, the green zone, is going to be segmented into 3 areas. This area inside this oval shape is near to the midline, therefore it is called around the midline. The term is para-axial. Axial means the midline and para means around. Then we have this segment between the 3 segments, and this is called the intermediate mesoderm, and the one to your left is the lateral segment of the mesoderm. We are in the subject of discussing the place where gametes develop, and this place in the middle or intermediate mesoderm, this is the place of development of the place that is going to house the very beginning of formation of the primary sex organs. It is called the Genital Ridge, and this is the place where it begins, and then the pink layer, the endoderm, is going to send cells into this intermediate mesoderm. This image is telling us few facts about the formation of the genital ridge, the house that the gametes are going to be formed. And let's ask a few questions. What is this structure inside this rectangle? It is the genital ridge. And what is this arrow indicating? It is indicating that the cells of the endoderm are going to invade the mesoderm and start collecting cells on the inside of the genital ridge. These things happen in week 4 of the beginning of fertilization. Now we are going to extract some knowledge out of this image. In week 6, that is after 2 weeks of the beginning of invasion of the epithelial cells of the endoderm into the genital ridge, they are going to divide and proliferate, and their origin is from the coelomic epithelium. epithelium lining the yolk sac. And what is the arrangement of these invading cells? They are in the form of cords, sex cords. Now, why are they called primary? Because they are the first to develop. Here is another image, and it tells a lot of facts. So let's ask ourselves a few questions. Now, we will start by noticing the genital ridge. Okay? And there is a genital ridge on the right and a genital ridge on the left. You remember the allantois is the little extension of the yolk sac into the umbilical cord, and the very beginning of the sex cells, the primordial germ cells, they start to differentiate and form around the allantois, and then they migrate. These are the primordial germ cells. They migrate. Look at the arrows. They migrate along the nearest structure to them, which is the large beginning of the large intestine. And once they reach the genital ridge, they go into the mesoderm of the embryo and start to settle down in the genital ridge between the primordial cords, on both sides, of course. Here are two figures repeating what we have just said in the previous slide. This is the primordial germ cells in the yolk sac around the allantois, and as they develop, they migrate along the nearest structure, which is the beginning of the large intestine, the third part of the intestine, which is called hind gut, and they move and they enter the posterior abdominal wall made of mesoderm, and they settle down in the genital ridge. Here is the yolk sac, and the primordial germ cells travel along the hind gut, the beginning of the large intestine, and they move around, and once they reach the midline, some will go to the right and some will go to the left and settle down in the genital ridge or the gonadal ridge. Let us check our understanding of the few events that we have just went through, and we were talking about primordial germ cells. First of all, we know that they give rise to the gametes. In the male, they form the sperms, and in the females, they form the ova. And we know for sure now they appear in the wall of the yolk sac near the allantois, and they migrate they travel along the allantois to the structure attaching the hindgut to the posterior abdominal wall called dorsal mesentery. Don't worry about this dorsal mesentery. You will learn about it several times in the future. And then they reach the gonadal ridge. And we have seen that. And they become associated with the primary sex cords. They live around them. And in the female, these cells become oogonia. They were primordial germ cells. In the female, they become what is called the oogonia. They develop a little step forward, and they divide by mitosis in the genital ridge during fetal life, and no oogonia are formed after birth. So that is a later event. Now, if the primordial germ cells fail to reach or to migrate to the genital ridges, then the ovary does not develop. It is the same story for the testis, the male primary genital organ. If the process is not complete and the primordial germ cells do not reach the genital ridge, the ovary or the testis, the primary sex organs, will not develop. Let's check our understanding again. Now, gonads begin development as what? As the genital ridges, one on the left and one on the right. These ridges, they develop during the 5th week of gestation or intrauterine life, and they come from intermediate mesoderm. Do we know what is intermediate mesoderm? The answer is yes. We have just described it as at one stage the embryonic disc was a single piece of paper, then it developed into 2, and then it developed into 3. The middle paper or the middle layer of the embryonic disc is a mesoderm, and the mesoderm divides into 3 segments, and you can refer back to the slide and see that the mesoderm is going to divide into 3 segments: the paraaxial, the intermediate, and the lateral. Now, there are epithelial cells of what? Lining the yolk sac. What is the yolk sac? The yolk sac is the cavity underneath the embryo, the future human being, and it is filled with nourishing fluid. These epithelial cells are finger-like bands, and these finger-like bands will get into the gonadal ridge, and then these finger-like bands are irregularly shaped primary sex cords. Why irregularly? We have seen the image. They are branching and occupying the genital ridge, and these are the primary sex cords. If there is a primary, then there would be secondary ones, but we are here at the stage of primary sex cords. These cords will grow into the genital ridge, and they form an outer cortex any outer layer of anything is called a cortex. And the inner medulla, the inside of structures. And this is the stage of indifferent gonad. What does that mean? It means that all the processes we have just described, they happen in the male and in the female. So the genital ridge now, at this point, is the same in the male and in the female. And it represents the place or the house that the primordial germ cells will come and live in it. Do you know what is this? This is the international symbol of female. After seeing the previous slides telling the story of formation of undifferentiated sex organ, which is the same in the male and the female, now the next short story is the story of developing this undifferentiated sex organ into an ovary in the female. This change from undifferentiated into the primary sex organ of the female, which is the ovary, is going to happen because of what? First, because of the X chromosome in the pair of sex chromosomes, and because of the genes and the hormones. And we will have development of the female genital system, which you can see in the figure to your left. Where is the female genital system? It's in the pelvis, and it opens into the outside through the vagina. Now, the vagina is a fibromuscular tube that connects the cervix to the outside. Now, details of these female reproductive system will be addressed later on when you take the urogenital system. Where is the next arrow pointing? It is pointing to a canal connecting the uterus to the vagina, and it is called the cervix. Then it is the uterus. The uterus is a central part of the female genital system, and it is a pear-shaped structure. The details will be postponed later on. But this uterus is the place of developing the embryo. Now, in this uterus, there is a cavity, and the cavity is lined with special cells called endometrium. This layer is going to receive and do the implantation of the embryo. The wall of the uterus is made of 3 layers. The inside is the endometrium. The muscular part is called the myometrium, and then the outer layer is covered by a special serous membrane called the peritoneum. Then the uterus is going to open into 2 tubes, one on the right and one on the left, and these are called fallopian tubes. Sometimes they are called the uterine tubes. And then we have the ovary. The ovary now is differentiated into a female sex organ, and it is the primary sex organ. Why is the word primary there? It is there because it is the place of developing gametes, developing sex cells. Now, what are the other parts called? The other parts are tubes, different types of tubes for the migration of gametes. Although migration of gametes is an important process, which we will give a summary of it later on. What is this figure? This is a figure of a cross-section of the ovary, the female primary sex organ. Now, what is this image telling me? First of all, it is telling me that the outer layer is the cortex. And in the cortex, we have the first, or the primary oogonium, the first stage of starting to develop into a mature ovum. And we will show you examples of this cell. Then the oogonium is going to develop by adding more cells on the outside, and this is the primary follicle, which is going to add more layers, and then it is called multi-layer primary follicle. And when the surrounding cells start to develop cavities, it is going to be secondary follicle, and these cavities will unite with each other and form a large cavity. And once we have a large cavity, then this is called Graffian Follicle. Now, as you can see here, this Graffian Follicle has a big cavity, and the surrounding cells, or the Granulosa Cells, are attaching the ovum to the periphery of the cavity, that's the antrum, and then as it gets larger and larger, it is going to stretch the outer surface of the ovary, and then it ruptures. The follicle ruptures, and the oocyte is released from the ovary. What is this image? You know, pictures talk 1,000 words, and we can extract what we want from this image. What is this structure inside this yellow oval shape? The answer is there are two things. We can see that there is a big cell surrounded by cells, and this is called the primordial follicle, the least developed follicle in the ovary. And the large cell inside this primordial follicle is the oocyte, the primary sex cell. And you can see that this oocyte is surrounded by dark sausage-shaped nuclei, and here it is a single layer, and these are called follicular cells. From where do follicular cells come? They come from the surrounding tissue to the primary follicle. The questions and answers opened our mind to notice lots of things. In week 10, this is what is going to happen. In week 10, we will find that the genital ridge is undifferentiated gonad. It is the same in the male and in the female. Then, because of several factors we have just mentioned, this undifferentiated gonad is going to go a step forward and becomes identifiable ovary because of the genetic of the X chromosome, because of hormones, and the primary sex chords, which we talked about, and they are the epithelial cells of the oocyte going into the genital ridge, and we talked why we call them primary, because they are going to change in nature. These primary sex chords will degenerate, they will die and disappear, and secondary sex chords are going to appear, and they are called now cortical chords, because they will not grow all the way, they only grow into the outer layer, and that's why they are called cortical. And these cortical chords, they come from the surface epithelium, the same origin of the primary sex chords, and they grow into the genital ridge, which is made of the mesoderm, the intermediate mesoderm, and the word mesenchyme is used as another term for the mesoderm. And when these secondary sex chords enter the genital ridge, they find that there are primordial oogonium, primordial female sex cells, while the primary sex chords, when they went in, it was only mesoderm. So the situation now is different, and the entry of these cortical chords, they are there to form a follicle which is called primordial follicle. So the primordial follicle, which is diploid in number, diploid is a term describing the number of chromosomes, and in diploid cells it is 2n. That means it contains the whole set of 46 chromosomes. These primordial follicles, they are made of an oogonium, the very early female sex cells, plus single layer of squamous follicular cells. We know a single layer is one layer, a layer made of one group of cells, and squamous means thin, and the follicular cells are the cells of the secondary sex chords that went in and divide in number and fill the space. And these cells making the single layer around the oogonium will start, with the help of the oogonium, to form a layer, a glycoprotein layer surrounding the oogonium and separating it from direct contact with the follicular cells, and this is called Zona Pellucida. Zona Pellucida has a story, and we will be talking about it later on. What is this view? It is a true histological section of the cortex of the ovary. What can I see in this section? If you notice the follicle inside this yellow circle, you can find a clear cytoplasm, whitish area, and the nucleus is in the middle. This is a very early oval cell, and it is surrounded by a single layer of thin cells. Thin cells are called squamous cells. If you ask yourself how many layers are there, the answer is a single layer. And here is another example in a nearby follicle containing an ovum and surrounded by a single layer of thin cells. You know it is a single layer because you can see these dark spots which represent the nuclei of the surrounding cells. These are features of what is called primordial follicles. These are the very early stages of development. When we go around and look for other follicles, we can see a follicle here. First of all, the ovum or the oocyte is larger, and we can see that the surrounding cells are not thin because the nuclei are not like a simple line. Nuclei of the surrounding cells are cuboidal, like a cube, and these are features of the next stage of developing primordial follicle, and now it is a primary follicle. Then you compare between an ovum surrounded by a single layer of thin cells and an ovum at the tip of the curved arrow. You can see that there is a difference between the single layer lining of a primary follicle and a single layer flat cells surrounding the oogonium. This is another histological section of the cortex of the ovary, and when we are curious, the questions will help us extract information. If we look at the large follicle in the middle, and we can see the oocyte, the primary oocyte. Now, why is this primary oocyte, and why is it a primary? Because if we can see this yellow structure is pointing at that the surrounding cells are cuboidal. They are not flat. Cuboidal means that the two dimensions of a cell are about equal, and this is the description of cuboidal cells. Then we go to the point of this arrow, and we can see a dark line surrounding the ovum, and this is glycoprotein material secreted by both the ovum and the follicular cells, and it is called Zona Pellucida. Now, for this reasons, this is a primary follicle containing primary oocyte surrounded by single layer of cuboidal follicular cells, and the Zona Pellucida has appeared. Let us check our understanding of a primary follicle and why it has changed its name from primordial to primary. We have noticed essential basic facts, which are that the oocyte is getting larger and it starts to have more than one layer of follicular cells. What is next is that these follicular cells, they become to be known as Granulosa cells. Why granulosa? Because they are starting to develop granules of hormones to be secreted. Now, these two layers become more than two, and they are multiple, and then the follicle is to be called multi-layered primary follicle. Now, the oocyte itself is arrested at a very early stage of first meiotic division, and that is the prophase. And do you know when is it going to complete this? It is going to complete this first meiosis after or beginning of puberty. So it is going to stay in this place, in this phase of meiosis for 12-13 years, and therefore it is exposed to chances of being changed. Then the Zona Pellucida. Zona Pellucida is a thick glycoprotein layer. It is situated between the oocyte and the zona glomerulosa, made of Granulosa cells, and it is in a stage of meiosis 1. This is a section of the ovary in which we can see what? We can see that the Granulosa cells are now multi-layers, and we can see that it is called multi-layered primary follicle. Now, what is the principle of this slide? This slide is for the sake of comparison. Comparison between what? Here we can see an oocyte surrounded by Zona Pellucida, and the Granulosa cells are multi-layered with large nuclei, and these are Granulosa cells coming from the surrounding tissues of the oocyte, and therefore this is multi-layered primary follicle. While if you go up, you can see that there is a primordial follicle where it is having smaller oocyte, and there is no Zona Pellucida, and it is surrounded by a single layer of flat cells. Let's ask questions about this section. First of all, the story of embryology is that it is made of sequence of events at specific times. Now, in this section, what kind of follicle is it? It is multi-layered primary follicle. But if we look at the tip of the yellow arrow, you can see that there is an attempt of forming a small space. This small space is going to change the name of this follicle. But this space is very small, and any space is called an antrum, and this is a small antrum. Therefore, this development is going to change the name of this follicle from primary follicle into secondary follicle. With the help of curiosity and asking questions, now we have a follicle in the center of this slide. Now, the little space we have just seen in the previous slide is getting larger and larger. And in addition to that, more spaces are developing in zona glomerulosa. And what do you think is going to happen next? This is a secondary follicle because of presence of vacuoles containing fluid in zona glomerulosa. Now let us collect some basic important facts about a secondary follicle. It is very similar to primary follicle, meaning that large oocyte and multiple layers of zona granulosa. Then they are larger. Secondary follicles are larger than primary, and they start to have more follicular cells. Follicular cells, you know, they have another name, and it is Granulosa cells. There is going to be small vacuole production, and these small accumulations of fluid in the intercellular spaces of the granulosa cells. These spaces, gradually, they are going to join together, meaning to coalesce to form a large cavity, and this cavity is called an antrum. Now, there are also surrounding granulosa cells, and these cells surrounding the oocyte are going to still connecting the oocyte to the granulosa cells. And this connection between the oocyte and the granulosa cells is called cumulus oophorus. Then things will develop. Now, the surrounding granulosa cells are now to be called theca. And there are two layers of the theca cells. The theca interna is a group of granulosa cells surrounding the Graffian follicle, the follicle that has a single antrum, and the theca externa on the outside, and it contains more fibrous tissue than cells. And the theca interna is going to secrete enzymes. Therefore, if we go back and remember that we called the follicular cells, which are growing in number surrounding the oocyte, they are going to start producing granules. Therefore, their name has been changed from follicular cells to granulosa cells. We need to ask questions. What is this section? Here we can see that the little spaces in zona glomerulosa has united together and formed a large single cavity filled with fluid. These cavities filled with fluid are important for the nourishment of the oocyte. So this is the last stage of developing follicles. This black arrow is indicating the oocyte, and this arrow below it is pointing at the granulosa cells that connect the ova or the oocyte to the granulosa cells, making the periphery of the antrum. This number of granulosa cells are called cumulus. What is a cumulus? It is simply granulosa cells of theca interna connecting the oocyte to the granulosa cells. And these are the features of the last stage of development of the oocyte with its follicles, and this is the Graffian Follicle. You know something, when you learn pieces of information and when you take a pen and a piece of paper and write what you have learned, this is going to be a very useful way of learning. We have just seen the Graffian Follicle, and we want to write down our observations. First of all, there is a single space. This single space is called the antrum. It is filled with fluid for the sake of nourishment of the oocyte. This antrum is surrounded by what is called membrana granulosa. It's a cover or structure surrounding the antrum and the oocyte, and these are granulosa cells. These granulosa cells surrounding the antrum and the oocyte, they project into the antrum, and this is called cumulus oophorus, which we have just seen, and they connect the oocyte to the surrounding granulosa cells. Now this Graffian follicle is getting larger and larger. Why is it getting larger? Because of more fluid is accumulating in the antrum. It reaches the surface of the ovary, and when it increases in size more than the cover of the ovary can stand, the Graffian follicle will burst, and the oocyte is expelled outside the ovary. This is ovulation. What comes out of the Graffian follicle is the oocyte, the Zona Pellucida surrounding it, plus the group of granulosa cells that are surrounding the oocyte. So there are three structures expelled at the point of fertilization. The corona radiata, they are no more than follicular cells. At fertilization, meiosis II is being completed. We need the aid and the help of questions. What is this diagram? This is the diagram of the expelled oocyte with its coverings. And let's review what are the structures surrounding an oocyte. It is corona radiata. These are granulosa cells on the outside, and then Zona Pellucida. And on the inside of Zona Pellucida, there is a thin layer of jelly material called vitelline membrane. And then we have the oocyte, cytoplasm, and the nucleus. Some people call the nucleus of the oocyte as germinal vesicles because it is going to divide and divide, and the repeated division is described as germinal, and the nucleus, the very important, now it is 1N, half the number of chromosomes. And with the aid of questions, number one, have we seen this section before? The answer is yes, we have. It is what? It is a diagram of a cross-section of the ovary, the primary sex organ in the female. At that time, we were discussing the types of follicles, and in the last few slides, we reached the stage of the oocyte being expelled. When these events are happening in the cortex, and at this event, when the oocyte is expelled, what remains in the cortex is the granulosa cells. These are hormone secreting cells, and they have granules. That's why they are called granulosa cells. These cells, they start to secrete hormones. In fact, they have started secreting hormones much earlier than this event, and it becomes functional, and this is what is called developing corpus luteum. This is what? This is what remains of the granulosa cells. And when it develops, it is well-established structure, and it is yellow in color, and this is the functional, the fully functional granulosa cells, and it is called corpus luteum. If pregnancy is happening, this corpus luteum is going to stay active and secreting estrogen and progesterone, promoting pregnancy. If pregnancy or fertilization does not happen, there is going to be no need for these hormones, and no need for the granulosa cells, and no need for the corpus luteum. Therefore, it is going to atrophy. That means it stops functioning, and when structures stop functioning, they get smaller and smaller, and this is the last non-functional corpus luteum, and it is called a scar, Corpus albicans. It is the degenerating functional corpus luteum. You know something, it is always useful to write down things, because when you write them, first of all, you test your short-term memory. Secondly, it elevates your level of understanding. And let's have some very basic facts about the corpus luteum. What is the color of the corpus luteum? It is a yellow color. How do I describe it? It is a glandular structure. What is it made of? It is made of active follicular cells. What is another name of the follicular cells? It is Granulosa Cells. Where is corpus luteum? It is in the cortex of the ovary. When does it form? It is formed when the Graffian Follicle ruptures and discharges the oocyte. If it is a glandular mass, what does it secrete? It secretes large quantities of what? Of pregnancy promoting hormones, hormones that act to make pregnancy continues. And what are these hormones? They are the estrogens and progesterones. Is it going to stay active and secreting? The answer is no, it depends. But when pregnancy does not happen, it is going to be atrophied. That is, it shrinks and degenerates into a white scar in the cortex of the ovary called corpus albicans. If pregnancy occurs, what is the situation? The corpus luteum, in case of positive pregnancy, it will continue secreting the pregnancy promoting hormones until parturition. What is parturition? Parturition is the process of giving birth to a baby. This is a group of laparoscopic photographs. What is laparoscopy? It is putting an instrument into the abdomen through an incision and shining a light and watching the structures live. And in this image, we can see the red bulging area is the very distended about to rupture Graffian Follicle, and the second image to your left, you can see that there is an oval yellowish structure protruding from a point. This is the oocyte coming out of the Graffian Follicle, and in this image the oocyte is about to be separating from the Graffian Follicle, and the lower right image is about the ovum or the oocyte is to be falling down from the ovary. This is a good practice for you to comment on, and I will help you during this process. which structure is the whitish large structure in the image, and what is this bulging reddish coming out of the surface of the ovary, and here is what is going to be expelled out. What is this structure? It is the oocyte about to drop out of the Graffian Follicle. I have put this slide for you to reactivate your knowledge of the different parts of the female genital system. Now, if you ask yourself questions and try to identify the following structures. What is this structure? You know this structure after the previous few slides. And what is the name of this structure? And these arms are coming from the last part of the fallopian tube or the uterine tube, trying to surround the ovary so that when the ovum is thrown out, these fimbria will catch the ovum and take it in. After the fimbria, there is this structure of the fallopian tube, and it is called the ampulla. In this place, the ampulla, fertilization will take place. Now, you may ask, fertilization is fusion of the oocyte with the sperms. How come that the sperms reach this area? This is going to be explained later. This is a diagram of the uterus to your right, and you can see the ovary, and in this area, inside this triangle, you can see that the Graffian follicle has ruptured and the ova or the oocyte is dropping down. This is a process that is very important because it has to be caught by the fimbria of the fallopian tube. If the fimbria of the fallopian tube are not functioning well, then this oocyte is going to drop down in the pelvis. And soon after the fimbria catch the ruptured Graffian follicle and catch the oocyte, the sperms will be arriving and fertilization will take place. What is this image? This is a three-dimensional image of the lining, the inside membrane of the fallopian tube, and you can see that when the oocyte or the egg or the ovum enters the fallopian tube, these extensions of the lining of the fallopian tube, they are called cilia, and they move, beating things from the ovary end of the fallopian tube to the uterus. This is a diagram of the uterus to your right, and you can see the ovary, and in this area, inside this triangle, you can see that the Graffian follicle has ruptured and the ova or the oocyte is dropping down. This is a process that is very important because it has to be caught by the fimbria of the fallopian tube. If the fimbria of the fallopian tube are not functioning well, then this oocyte is going to drop down in the pelvis. And soon after the fimbria catch the ruptured Graffian follicle and catch the oocyte, the sperms will be arriving and fertilization will take place. What is this image? This is a three-dimensional image of the lining, the inside membrane of the fallopian tube, and you can see that when the oocyte or the egg or the ovum enters the fallopian tube, these extensions of the lining of the fallopian tube, they are called cilia, and they move, beating things from the ovary end of the fallopian tube to the uterus. Here is a diagram of what happens in cases of development of the oocyte. It starts as oogonium, and it has full set of chromosomes, then gets larger and surrounded by follicular cells. It is called primary oocyte. This primary oocyte, which is 2N, having full set of chromosomes, meiosis 1 is going to start here, and meiosis 1 is going to stop at prophase. And then when reaching puberty, the primary oocyte will change into secondary oocyte by changing the shape of the follicular cells, and it is trying to become 1N, and here meiosis 2 is going to be completed at puberty before fertilization. What is this? This is a mind map. What is a mind map? A mind map is the most powerful thinking tool present in the 21st century. What does it do? It presents the key words, basic facts, and integrate them in their right place in an A4 piece of paper. Now, it is the best way to test yourself. If you can mind map it, you know it. If you cannot mind map it, you don't know it. When you don't know it, you go and learn it. So it is a very good way of telling you the truth. You know this and you don't know this. Therefore, you go and remedy your knowledge. This is a mind map of oogenesis, which is the process of formation of oocyte. Where does it start? It starts its story in the yolk sac, and we have went through this information, but we are summarizing it and putting things in its right place. That is integration. What do we know about the yolk sac? It is an extra embryonic cavity inferior to the embryonic disc, which is the future human being, and in the yolk sac there is production of primordial germ cells. The next idea is that there is a structure called Genital Ridge, which is a very early ovary. It is the skeleton of the ovary. What is the basic knowledge of the genital ridge? The genital ridge is a shelf-like structure medial to the future kidney, and it is higher up in the posterior abdominal wall, and it is going to house the primordial germ cells, which are called oogonia. These cells, the oogonia, they are going to multiply immensely by mitosis, and some cells will get larger, and when these cells get larger, they are surrounded by a group of cells called the follicular cells, which they come from the ovary surface. When an oogonia is surrounded by follicular cells, it is a primary oocyte. This primary oocyte is going to start meiosis 1, but stops at prophase and continues after puberty. Then there is more mitosis until a huge number is achieved, approximately 7 million primary oocytes present in each ovary. Now, this is going to stop, and primary oocytes are going to die, large number of them, and this death is programmed in the genes, and it is called apoptosis. What is apoptosis? It is programmed death of cells. Then we come to 5 months while the embryo is inside the womb. During this age, most of the primary oocytes are dead, and by the 7th month, the number is reduced, and these structures are near the surface of the ovary. These ones are going to survive, and these are primary oocytes that remain alive, surrounded by follicular cells, single layer of follicular cells, and they are called primordial follicles. And by the 7th month, there are about 2 million. After birth, everything is going to stop and rest. This stage is called diploiteen stage of standstill. After birth, there will be the childhood, and during the childhood, more death of the primary oocytes and the oogonia. And these primordial follicles are going to be sacrificed more and more. Then comes puberty at the age of the common age of 12 or 13, sometimes earlier. There will be the stimulus of hormones. To what? To the follicles. What hormone? The hormone is the follicular stimulating hormone, a very descriptive term of what this hormone does. And when these follicles are stimulated, 5 to 15 of the primary follicles are developed at a time. And at this stage, at puberty, there are around 400,000 primary follicles. Then, when the primary follicles receive the stimulus, these primary follicles contain primary oocyte and follicular cells around them, several layers of the follicular cells, and they get larger. Then comes the secondary follicle, which is larger than the primary, and the secondary is when the granulosa cells or the follicular cells develop cavities, and a single cavity is called an antrum, and then they unite to form one large space which is filled with fluid, and there is the formation of Zona Pellucida, which started at the primary follicle, and it continues the process of formation of the Zona Pellucida. Now we're finished with the secondary follicles, and we come to the Graffian follicles. These are called Graffian follicles because they get larger, more fluid in the antrum, and it is about to ovulate, about to get out of the follicle. These cells, the Graffian follicles, are going to resume meiosis 1, which was stopped at the early prophase many years ago. And when meiosis 1 resumes and meiosis 2 starts, the division is going to result in one daughter cell and a polar body. What is a polar body? A polar body is half of the chromosomes with little cytoplasm thrown away. So the 2N cells at this stage, they become 1N. And the Graffian follicle is going to start meiosis 2 and also stops at anaphase, and it is ovulated at the stage of prophase of meiosis 2. Once it is ovulated and thrown out, the oocyte dies within 24 hours. This is the international accepted symbol of a male. This image, which you have seen at the beginning of the story of formation of the genital ridge, and which will result in the formation of undifferentiated gonad, undifferentiated genital ridge. It is going to involve what? You start with the early embryo. There's a cavity above it and there's a cavity below it. This is the yolk sac, and then the yolk sac has an extension called the urachus, and the primordial germ cells develop here as 2N cells. Then you go to the right and you follow the process of migration and then inhabiting the genital ridges. This is part of the story you have heard before. You also know this story because you have heard it before and you can describe it yourself. It is a kind of a practice of your recent memory. Questions will help us understand what is going on. This is a true histological slide in which it shows that a seminiferous tubule, which is the group of cells producing sperms, the male gametocytes, and it is surrounded by an outer layer made of several structures, fibrous tissue, and muscle cells. And then you have this layer of group of different stages of male gametes, and this layer is called the germinal layer. We know what germinal means. It means the dividing process, and in the middle you have a lumen. This arrangement is different from the ovary. The ovary has cortex and medulla. The testes, the seminiferous tubules, have an outer shell, a germinal layer, and a lumen. This is a diagram of the testis, and it has different structures, quite different from that of the ovary. The ovary has a cortex and a medulla, and follicles develop, and the ovum is thrown out of the Graffian Follicle. Here the testis is different. The testis, as a primary sex organ of the male, they have lots of ducts, and sperms are carried into these ducts for a long distance, and it takes a long time. Where is the testis? Testis normally is at the bottom of the scrotum. What is the scrotum? The scrotum is a pouch of skin in the perineal area, just posterior to the penis. And structures coming in and out of the testis, whether arteries, veins, ducts, this structure is going to pass from outside the body that is from the scrotum to the inside, and this is called the Spermatic Cord. The first figure here, the structure colored green is the outer shell of the testis, and it is white fibrous tissue. Therefore, it is called tunica albuginea, the white layer. Then, in the next image, this tunica albuginea will send septa, partitions into the inside of the testis, and these are fibrous septa. This figure is showing us that the green area are incompletely formed lobules created by the fibrous septa. So these are the lobules. There are 200 to 300 lobules in a single testis, and they are filled with, each lobule is filled with 1 to 4 seminiferous tubules. Seminiferous tubules are not straight, they are tortuous, they are twisted, therefore they are called convoluted. Next to the seminiferous tubules, sperms are collected into the straight part of seminiferous tubules, which is called the straight seminiferous tubules. Next, the straight seminiferous tubules are connected to a network of ducts in the hilum or in the center of the testis, and these are called reti testis. Next is from the reti testis, now there are ducts that will take the sperms away from the testis, outside the testis, and these are called efferent ductules. There are 12 to 20 of them. Next, when the efferent ductules come out, they start to form a single duct. This single duct is 6 meters long, and it is convoluted on itself in a very compacted manner. And the first part of it is called the head of epididymis, and then the middle part is called the body of the epididymis, and the last part is the tail of the epididymis, and then the convolution disappears, and a duct is continuous with the tail of the epididymis, and this is called Vas Difference or ductus difference. So you can see that when a sperm is formed in the seminiferous tubule, it is going to travel through a series of ducts. And the vas difference will go to the spermatic cord, and the spermatic cord is going to pass through the abdominal wall to enter the pelvic cavity. And the next is the ejaculatory ducts, the ducts that are going to deliver the sperms to the urethra. These ejaculatory ducts, they are connected to 2 glands, one on the right and one on the left. These glands secrete fluid material rich in glucose, and you must understand that these sperms, they swim in a fluid, and the fluid is being added, and at the end of the journey, the seminal vesicles are going to add more fluid to the medium in which the sperms swim in, and then the ejaculatory duct is going to open in the prostatic urethra. So the male genital system ends in the urethra. The urethra is going to be a common passage for urine and seminal fluid containing sperms. This figure of the testis and the ducts related to the testis is intended to be left to you to practice your knowledge so that you can confirm and remember and understand the testis. You are required to name the structures that are marked with the red arrow, then with the black one, and here is the epididymis. This word is a bit tricky in writing it and in pronouncing it. That's why I have already put it down. And this is what? What is this? This is a diagram of the testis, and between these two lunar figures can see a convoluted seminiferous tubule, because we are going to study histological sections of this seminiferous tubule. What is this? This is a histological real section of the seminiferous tubules, and you can see that the sections are packed together. Why are they packed? Because the seminiferous tubule is very much convoluted, and you can see that each tubule has a lumen and a group of cells at the periphery of the lumen. These cells are the developing sperms starting from spermatogonia, like the oogonia, and different stages of development till you reach the lumen where the sperm is produced. Now, in the male, you need a hormone called testosterone, and testosterone are secreted from cells outside the seminiferous tubules, but inside the lobules. They are between seminiferous tubules, and they are large cells, and they stain differently, and they are called Leydig cells. Another name is interstitial cells. With the help of questions, this is a closer view at the outer shell of the testis, which is made of connective tissue and muscular cells and other cells, which we will talk about later on. And this is the white layer surrounding the testis. It is tunica albuginea. Then we have a cross section, different cross sections of the seminiferous tubules. Now we will have a closer look at the germinal layer, the cell that are in this layer, which tell the story of the beginning of the spermatogonia up to the formation of spermatids and sperms. The story starts with the wall of each seminiferous tubule, which contains smooth muscles, fibrous tissue, and blood vessels and other types of cells. This is going to surround the section of the seminiferous tubule. Then at the periphery, if you look down to your left, you will find cells near the periphery. These are small cells with dense nucleus. These cells are spermatogonia, like oogonia, the first stage of development. Then the next stage is this group of cells starting at the bottom. They are larger, and this last cell, you can find that the chromosomes are dispersed because these cells are dividing very rapidly, and they are next to the spermatogonia, and they are the primary spermatocytes. These primary spermatocytes will develop into secondary spermatocytes, and these secondary spermatocytes are 1N cells. They contain half of the number of chromosomes, and they are difficult to see because they divide very quickly. The process of division of cells producing sperms is very fast because the process needs to produce millions of sperms. Therefore, the activity of mitosis and meiosis is very energetic. Then we have a story. This story is that of the spermatogonia, primary and secondary spermatocytes. They need feeding, and this feeding as a source of energy and taking care of is provided by a large cell. This large cell is called Sertoli Cell or interstitial cell. This is the nucleus of this cell is faint and large, and therefore it can be recognized very easily. Now look for the circles which would appear now. This is a nucleus of Sertoli cells, and if you look up and to the right, you can see two more nuclei. Then secondary spermatocytes are going to develop into spermatids, the early stage of mature sperms. The nuclei of these cells is near to the lumen. They are small and condensed, having a dark color. These are two nuclei of two spermatids. These are another two nuclei of spermatids. This is a diagram of what is happening in the germinal layer of seminiferous tubules. To start with, there is a basal lamina. Why do we have basal lamina? Because these gametocytes are of epithelial origin, and every epithelium should have a membrane that makes a base for these cells. So this is a basal lamina, a basement membrane. Then look at this cell. It is the Sertoli cell. It is a large cell with large nucleus, and the chromatin of the nucleus is different from the arrangement of other gametes, and it extends from the basement membrane to the lumen, and it is wide cell as well. And you can see that the gametes are getting into and attached to the outer membrane of these Sertoli cells. Now, to your right, the cell sitting on the basement membrane is the spermatogonium. This is what comes from the yolk sac and lives into the genital ridge. This is the very primitive sex cells. And it is going to develop into primary spermatocytes, and this is getting larger, and it is dividing very energetically. Then, next to the primary spermatocyte is this group of three. They are secondary spermatocytes, and these divide very quickly, and they are difficult to see in sections. Then we have the early spermatids. These are the very beginning of the formation of the sperms. Then these spermatids are going to develop into sperms. If you look down, you can see that this red arrow is indicating group of cells outside the seminiferous tubules, in the space between the tubules. These are called interstitial cells, and these are the cells that produce the testosterone. And testosterone produced is being picked up by capillaries. If you look at the green arrow. Then this is a figure of meiosis happening in secondary spermatocytes. Then when the spermatids are formed, the process of spermatogenesis has ended. Now a new process is going to develop, and this is going to be called spermiogenesis instead of spermatogenesis. Spermiogenesis is modifying the spermatid into fully functional, fully formed sperms. These blue structures attached to Sertoli cells are late spermatids, which are nearly gaining the adult shape and function. Now, with the help of the questions, which are the main motivation for learning, what is this section? This is a section in a seminiferous tubule. We can see the periphery at the bottom of the slide, and we can see the different types of cells producing the sperms. If you look at this cell inside this black circle, you can see that the nucleus is large compared to the dark areas of the cells around them, and the cytoplasm is pale and the nucleolus is visible, and this is Sertoli cell, the cell that feeds, or the cell that nurses the different stages of developing sperm. Question: have I seen this section before? Yes, it is just like the previous one, and it has the same description. But if we look into the inside of this black circle, we can see a cell near the outer limit of the seminiferous tubules, and it has dark stained nucleus. These are criteria that will lead us to conclude that this is spermatogonia, the very early stage. Now we know where we are, and if we look at this cell, we can see that it is in the middle way of the germinal epithelium, and the nucleus has chromosomes that are fragmented, and you can see several of them, and these are the criteria that would lead us to identify this cell as primary spermatocytes. Why we almost, whenever we look at primary spermatocytes, we see this kind of chromosomal arrangement? It is because this cell is undergoing very energetically into mitosis to produce millions. With the help of asking questions, where is this section? You can see that in the center there is a kind of a triangle, and you can see that there is a germinal epithelium on your right, germinal epithelium on the inferior side, and one on the top. This is an area between seminiferous tubules, and these cells are the cells of Leydig, which are producing the testosterone. This is the same figure we have seen 2 or 3 slides before, and we talked about each single type of cells, and this is a test for your knowledge. You can identify a cell and you know what is the name, although it is labeled, but it can be a useful exercise. After formation of the spermatids near the lumen of seminiferous tubules, there is a new process which is going to change the shape of the spermatids, and this is called spermiogenesis. Generally speaking, what is spermiogenesis? It is changes that transform spermatids into a new adult sperm called spermatozoa, another name for sperm. Generally speaking, the process involves discarding excess cytoplasm and growing tails. Now, when you go to figure 1 to your right, the cell is rounded. It has the organelles that are important for maintaining the cell, and there is a very important development of very important structure, and that is the acrosomal vesicle. This acrosomal vesicles is going to be very useful in the process of fertilization. When you go to figure 3 and 4, there is the development of flagellum. Flagellum is very important because it is the motor for the movement of the sperms. Sperms travel in the female genital system for a very long distance in relation to their size, and the flagellum should be anchored in the cell, and the anchoring structure is the basal body. Now the flagellum is going to get longer and longer, and if you go to figure 5, the acrosome has developed very well on top of the nucleus. The nucleus is going to contain the male sex hormones, the 23 pairs of the chromosomes, and then because the sperm is going to need to swim very fast and very long distance, so we have a collection of mitochondria as a source of energy, and the excess of the cytoplasm is discarded away, and then we reach the final shape of the sperm. If you look at the top red arrow, you can see that it is pointing to the acrosome. This is a very important structure. Then lower down is the nucleus. The nucleus is holding the genetic material, the 1N, the haploid number of chromosomes, and then we have below the head we have what is called the middle piece, which contains lots of mitochondria, and then the tail. The tail is a long flagellum. This is a 3D image of a well-formed sperm. It contains three main areas: the head, at the tip of which is the acrosome, and then the nucleus, which contains the genetic material, and then the middle piece is the area of condensation of mitochondria, and then we have the flagellum. Test your knowledge on this diagram and go through the naming of the structure and see which one is having the acrosome, which one is the mitochondria, and the parts of the tail. Here is an outline of the male genital system. Now, this is the testis, the primary sex organ in the male. Everything else is a group of passages, group of ducts, and the testis is at the bottom of the scrotum, which is a pouch of skin hanging down in the perineal area. Now, what does that mean? That means the testis is outside the abdominal cavity. Why is that? Because the process of spermatogenesis and spermiogenesis require lower temperature than the core temperature, the temperature inside the body. The ovary did not need that. Then, after the testis, we have the epididymis, and we had a look at the epididymis in details. And then the sperms will, after finishing the journey in the epididymis, which takes a long time, and it is developing while it is in the epididymis. The spermiogenesis, especially, most of it takes place in the epididymis. Then we have the Vas Difference, and the Vas Difference, or the ductus difference, is going through the spermatic cord, and it is going to enter the pelvic cavity. And then it turns around on the lateral wall. It goes behind the urinary bladder, and it is going to entering the prostatic gland, and this is called the prostatic part, or the ejaculatory duct. At this point, the seminal vesicles, two glands at the back of the urinary bladder, are going to add secretions to the seminal fluid, and then they enter the prostate gland as ejaculatory duct. And at this point, the genital system is going to join the urinary system. What is this? This is a mind map telling the story of spermatogenesis and spermiogenesis. If you look at the center of this mind map and then start at the black arrow, the story starts in the yolk sac. This is a repetition of what happens in the female as well to form the undifferentiated gonad. And then these primordial germ cells, they will come and live in the genital ridge. The genital ridge, if you go down, it is not going to stay in the abdomen, near the kidney. It is, in the case of the male, it is going to travel along the posterior abdominal wall, then along the gap in the abdominal muscles, and end up at the bottom of the scrotum. It must do that. Otherwise, spermatogenesis will not happen. Now, at birth, what's happening at birth? At birth, the testis must be at the bottom of the scrotum. If it is not, then it should be brought down by surgery. And at birth, the primordial germ cells are called spermatogonia, and they are in the cords of cells. At birth, seminiferous tubules don't have a lumen, but they have the Sertoli cell, the nursing cell for these spermatogonia. Next is childhood. From the moment of birth till puberty, nothing happens in the testis. It stays as we have just described. Now, in puberty, hormones come into action, and it is the follicular stimulating hormone coming from the pituitary gland and stimulates and wakens up the process. Now, first of all, the seminiferous tubules, they start to have a lumen, and the spermatogonia inside these seminiferous tubules, they divide, and they collect in two groups. Type A, they start dividing and dividing and dividing so many times to produce millions of spermatogonia. And these millions are present in a functional area. They are reserve of stem cells, because oogonia are stem cells. Then this reserve will start, under the influence of hormones, to give a group of spermatogonia called type B. Type B, which are spermatogonia, they start to develop into primary spermatocytes with complete set of chromosomes. Now, these primary spermatocytes will go into meiosis 1 and stop at early stage, and it is going to be a very long prophase, and it's going to take 22 days. The primary spermatocytes are going to change into secondary spermatocytes after completing meiosis 1, and the result is half set of chromosomes, and then it is going to complete meiosis 2, and then when secondary spermatocytes divide to form spermatids, the spermatids are not separated like the nuclei. The spermatids have connected cytoplasm in groups of 4. And these groups of 4 are going to reach the lumen. All these stages are nursed by Sertoli cells. Now, once they reach the lumen, spermatogenesis has ended and spermiogenesis begins. When spermiogenesis begins, cytokinesis, which is separation of the dividing cytoplasm, is completed, and there is condensation of the chromatin of the nucleus, and there is acquiring the final shape of the head, a little constriction under the head called neck, middle piece, and tail, and shedding most of the cytoplasm. A very important structure on top of the head of the sperm is the acrosome, group of enzymes that are going to help sperm getting through the outer coat of the oocyte and to inject the nucleus into the oocyte. Now, this process from the oogonium to formation of mature functional sperms takes 64 days. And once the sperms are ejaculated, then they can be living for around 3 days. When the sperms are ejaculated into the vagina of the female genital organs, they are aiming at, and their target is reaching the oocyte, the ovum, at the ampulla of the fallopian tube.